Now, within RNA in particular, but also single-stranded DNA, a type of sequence called a palindrome can affect the overall structure of a RNA or DNA sequence. A palindrome is a word or a phrase that is the same when you read it backwards or forwards, and there are some examples below. Civic, you can see CIVIC, and if you go the opposite way, CIVIC. Race car, rotor. This Finnish word that I will not attempt to pronounce, but it means soap cup batch trader, is a palindrome. And also an ancient Greek phrase, again noted here, that means wash the sin as well as the face. Again, if you isolate these and read them from front to back or back to front, the actual sequence of the lettering is the same. We can describe segments of DNA or RNA as also being a palindrome. Now, in a DNA sequence, to be a palindrome, it means that the full read from 5' to 3' must be the same. So remember that if we're looking at a sequence, this might be the 5' end, this is the 3' end, but that means because these bind anti-parallel, the 3' end would be here on this strand and 5' here. So you can see to be a true palindrome, as you read from 5' to 3', or this way from 5' to 3', the sequence reads the same. Don't confuse this with something like a mirror repeat, which also is important structurally, but distinguished by the fact that within the same strand, you have a repeat when read from left to right or right to left. So here you can see T and T, 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 A, etc. So within one strand, if you have a complete repeat of sequence, this is a mirror repeat. But if you can read it from 5' to 3' on one strand of the DNA, and it is the same as the 5' to 3' of another strand of complementary DNA when read, that is a palindrome. Now you can see that these palindromes, because they have complementary segments, end up forming three-dimensional structures when they are just a single strand called hairpins. They can bind in on themselves. So for example, this palindromic sequence portion here has complementarity where this segment and this segment along the sequence are complementary to each other. That makes it likely that they can form hydrogen bonds if folded in on itself, as shown below, forming, again, what is called a hairpin. In single-stranded structures, and in particular RNA, this can occur a lot, and it will form very unique and sometimes complex structures. RNA, thusly, should not be limited to thinking of it as just one single vanilla strand, but instead as potentially having these very complex three-dimensional structures like proteins. And because of this, RNA molecules have also been seen to be involved in things like enzymatic processing of reactions. This three-dimensionality of them allow them to fold in unique ways, like we saw with proteins. Here you can see some three-dimensional RNA strands, again, folded similar to what you saw in proteins when our nucleic acids fold into three-dimensional structures. These are again stabilized by hydrogen pair interactions that are not occurring just between bases, but occurring not only between base pairs, but within other base pair interactions. These are non-Watson-Crick base pair interactions, meaning they don't form in the typical way where we just have A and T and G and C binding with each other, but they're forming unique hydrogen bonds in an overall way to fold them into three-dimensional structures. Again, these type of RNA molecules have been involved outside of just the processing of DNA to RNA to protein, but also in roles of things like enzymatic catalyzation of reactions. Here's where we also see some of these unique bases coming into play. 7-methylguanine, for example, which is a base pair that we don't find in DNA, but it can be involved in the folding and stabilization of RNAs like this top one, which is called the hammerhead RNA. Also, this N2-dimethylguanine, another unique base that we talked about, which is found in a subsequent three-dimensionally folded RNA, single-stranded RNA structure.